research, and I don't really know how I used it in my research. And
does anyone ever know?
so he gets mad because I don't know how to chi-square.
Yeah, I know. Yeah, on the reference slide.
But you use it, that's all that matters.
References will be put in.
And like during my presentation, I was like, I used a chi-square test to test blah blah blah. What is that? But how? And I'm like,
I can't do it.
I don't know.
I was like, what? I don't think it's gonna be on there, but he signed
It's
it.
magic.
What's that? Oh, okay. Never mind. Done?
No.
Yeah, okay. Let's see. I think that's about it. Some people are absent today for various reasons. But I want to tell you about something. How many of you are biology majors or intending to be biology majors? Raise your hands, okay. Biochemistry? Chemistry. Okay, and Brooks is data science. Is there anybody who, yeah, we're using psychology. Oh, psychology. So if you are a biology major, you make an application to the major. I'm not sure what they do in biochemistry and chemistry, if they do a similar type of thing. When you came in as first years, you probably declared yourself a biology major. But just because you declare yourself a biology major doesn't mean that you're eventually going to graduate with a biology degree, because you might not have grades that are C or above in your courses, et cetera, et cetera. So this is a time period when we want to basically just check in with you to make sure that you're kind of on track through the bio major. You would have to check with chemistry to see if biochemistry and chemistry are the same way. So there's an application. It used to be a Word file that we would send you out. I've been working on this week on getting a form, an online form that you can fill out and submit. But it asks you for the courses you've taken, the grades that you've gotten in those courses, because all of you have different academic advisors and things like that. It's just easier for us to have you report that all to one spot. The other thing that you need to be aware of is that for biology, biochemistry, and chemistry, you're all required to do undergraduate research. And so there are also questions on there that are new on this year's version of the application to ask you questions about who you might be interested in working with. Have you talked to that person yet about working with them next year? Because next year you'll start undergraduate research with one of the faculty members. You can do research with members in chemistry or biology, it doesn't really matter. But it's best if you find somebody who is doing the kind of work that you would like to be doing. And now is the time to start talking to faculty about that. Do not put it off until next fall. If you put it off until next fall, you're already behind the 8 ball, because you're supposed to be working on developing a project proposal and things like that in the fall. So you need to talk to faculty about what their research is involved in if you don't already know that. And you definitely need to talk to them about joining their lab. Simply because you want to work with a particular faculty member, they might not have enough space in their lab for you because they already have 6 or 7 students. And so by the end of the semester, you need to have talked to faculty and gotten it worked out that they know that you want to work with them, you know that they have space for you in their lab, and you've reached an agreement that basically says, yes, next semester I'm going to be working with you. You have to enroll in undergraduate research courses, and if you're a bio major, until you have submitted that application and we've gotten a chance to review your grades and things like that, we won't allow you to enroll in the research courses in the fall of your junior year. So that is that. Speaking of research, why do we have you do undergraduate research at William Jewell College? This is the part where you provide hypothetical answers.
Some experience for grad school.
Okay, so if you're planning on graduate school, it'll give you a good experience where that'll give you a taste of research. But of course, a lot of you aren't planning on going to graduate school. Why would we require it of those of you not planning on going to graduate school? so it certainly gives you hands-on experience. There's a variety of different ways to learn about science, one of which is sitting in a room, letting somebody talk at you all day, and hope that you actually learn something about science. Another way is to actually get involved in the process of doing science, and you learn about science in a different sort of way when you engage in science in that way rather than just book learning. Other reasons why we might require you to do undergraduate research. Are you guys just ready for spring break?
Yeah.
Okay. Any other reasons why we might require this of you?
Looks good on resumes.
Actually, it does look good on resumes, but that would be a really stupid reason to require you to do it. It's extremely time consuming and extremely costly, and so yeah, it does look good on a resume, but yeah, that's probably not the best reason for it. Now, when you go to medical school, so you talked about graduate school, when you go to medical school, we hear from students who go off to medical school interviews, and they talk about how the faculty that they were interviewing with got really engaged with them when they asked them what they were involved in with their undergraduate research. So not only does it look good on a resume, it also it gives you something to talk about with interviewers and things like that at medical school interviews that kind of sets you apart from other graduates from other places, because there you are talking about, doing flow cytometry or doing whatever it was that you did here, and it gives them the impression that, one, you actually know what you're talking about, and two, that you've got a good set of experience that's going to suit you well, whether you're in medical school or graduate school or whatever else. So there are lots of good reasons to do undergraduate research. Not a lot of schools do it, and of those schools that do it, oftentimes it's a very limited experience, because either not everybody gets to do it, or if you do it for a relatively limited time. You're going to span 2 years of working on this with a faculty member. I bring this up for two reasons. One, because I'm going to be sending out probably over spring break the app to the major. I'm going to send it to everybody in class. If you're not a bio major, just ignore the email. If you are a bio major, click on the link and go and fill it out. So I was already planning on talking about this, but as it turns out, I got an email today from Mike Strickland, a former Jewel graduate who is now a faculty member at the University of Idaho, and he just got what's called an early career grant from the National Science Foundation. They don't give out very many of these. It's a very large grant. It's $651,000. He was one of my research students. He worked with me for a couple of summers in Puerto Rico, and he was interested in decomposition, so we designed a decomposition project for him, and he continues to work in decomposition to this day, strangely enough. But when you're working in Puerto Rico, you don't have a lot of the infrastructure that you might have in Liberty, Missouri for doing research, and so he wanted to look at how invertebrates were affecting decomposition rate. This has nothing to do with genetics, as everything we do with research. So just humor me for a few minutes. So we had to build these mesocosms, these small communities that we built up from scratch. We had soil in there, we had dead leaves in there, and then we stocked them with different kinds of litter invertebrates, because litter invertebrates were thought to be important in determining decomposition rate, because they actually consume dead leaves in many cases. But being in Puerto Rico, we didn't have a lot of the things that you would normally rely upon, and so we went to the hardware store and we bought a bunch of underbed storage boxes. We used weather stripping from the hardware store to seal them up so that the bugs couldn't get in where we wanted them to not be, and the ones that we wanted to be there couldn't get out. We cut holes in the lids and glued plankton netting over the holes so that water could get in and so that water could drain out, et cetera, et cetera. There was a lot of kind of problem solving that went on with the work that we did in Puerto Rico, because we were doing work in a place where you had to be clever and creative in getting the research done. And it was kind of fun. It was a lot of work. And when he got back from those 2 summers of working there, he was applying to graduate schools. He got an interview at the University of Georgia, which is one of the premier ecology programs in the world, in reality. Possibly part of the reason that he got in was because he met one of the faculty members at Georgia while he was in Puerto Rico working, because there were a number of University of Georgia faculty who worked at the same site that we worked at. But while he was there was where he really kind of shined what doing undergraduate research does for you. So he was taking a tour of the labs, and he went to one lab where they were doing soil decomposition studies. And they had created mesocosms using PVC pipes, which is not unusual for ecologists to use things like PVC pipes. And what they did was, in this PVC pipe, they basically built a soil profile, a meter deep soil profile, and then they were inoculating this with different types of microbes, different types of soil microbes, to see how microbe community structure influenced the rate of decomposition, the rate at which microbes process the carbon that is in soil. And normally what you would do when you do this is you would put a cap over this. The cap has a little nozzle in it. You put a hose over that nozzle, you connect that to a syringe, and you suck air out of this headspace up here, and you suck the air out of there, and because decomposition has been going on, decomposition breaks down glucose and produces carbon dioxide, you can compare the amount of carbon dioxide in this headspace to the amount of carbon dioxide that was in the atmospheric air before you put that lid on top of it, and you can see how much carbon dioxide is being liberated per unit time, and calculate a decomposition rate for that. But the problem with this is that as they created these soil profiles in these PVC pipes, they watered them because microbes need to stay hydrated in order to survive, and some of them settled more than others. And so every pipe was slightly different in terms of the depth of the soil profile. And so now you can't just put a lid on top of there, because in order to calculate how much CO2 is being liberated, it's the amount of carbon dioxide per unit volume. And so one of the things that you need is a constant headspace, so that you can make this calculation. Well, you could actually measure the depth of each of these and have a headspace for each one, but the next time you go and water it, that headspace is going to change as the soil profile continues to sink. And so they were telling me about this project, and they were talking about how their challenge right now was how to get a constant headspace. And it took him just a couple of seconds to say, do you have a beer cup? And they were like, what do you mean a beer cup? And he's like, a red solo cup like you would have at a kegger. And they're like, oh, we can probably dream one of those up for you. And so they sent a graduate student off to find a red solo cup. They gave it to him. He took the red solo cup and he slid it right down in the tube, and it fit pretty well. There was a little bit of space around it. But he said, there's your constant headspace. Just put your attachment for your syringe on the bottom of it. That then allows you to suck air out, and whatever the volume of the red solo cup is, that's your constant headspace, and it'll be your constant headspace regardless of how much you have settled here. And he said there's a little bit of space between the inside of the PVC and the outer rim of the cup. But he's like, you can just go to the hardware store, get some weather stripping like we used in Puerto Rico to seal up my mesocosm boxes. Just wrap that around. That'll take up the rest of the space. Headspace issue solved. Where did he learn to do that? To quickly find a simple, exceedingly cheap solution to a problem that was vexing the people at this lab in just a few seconds. He could do that because he had spent 2 summers solving similar kinds of problems in association with his own undergraduate research.
Critical thinking college.
It's, well, we didn't even think about it as that. This is just solving problems. Can you think creatively? This wasn't critical thinking as much as it was kind of creative thinking. Oftentimes if you ask people if science is a creative activity, you know what they will say? No, creative activities are like art and music and literature and things like this. Science is a very creative endeavor because you sometimes have to think creatively about things to make any progress. One of the reasons that we have you do undergraduate research is because it trains your mind in a way that you're never going to get sitting in this class listening to me drone on about genetics. It's going to give you a way of thinking that is distinctive, that allows you to go to a place, figure things out. He's pretty convinced to this day that that's what got him into Georgia. A beer cup and a piece of PVC. I didn't teach him any of that. He learned that along the way as a result of doing undergraduate research. These are the reasons that we have you do it. It's sometimes scary. It's certainly a ton of work. But this is how you learn science in a way that you're never going to learn just sitting in a class listening to faculty lecture at you, or even sit in a class and do group work where you're figuring things out for yourself and making models of transcription and translation and things like that. Those are all good ways of learning also, but undergraduate research is going to teach you your science in a way that you could not have conceived of it without doing that. So, Strickland just got a big grant, and regardless of what you do, whether you end up going into academia or end up going into industry or medicine or anything else, being able to think in these ways is important. My girlfriend's father is suffering from septicemia because he got a pacemaker put in him. He's in the hospital. He's been there all week, which is why if I'm a little sleep deprived, it's because I've been spending most evenings up at the hospital. But when he goes in and gets his fillings, gets dental work done, they have him go on antibiotics the week before he goes in because he's on Coumadin, and that's a blood thinner, and so he's more prone to getting infections and things like this. When he goes in for dental work, they have him do that. When he went in for his pacemaker, they didn't have him do that. In spite of the fact that they're actually cutting a hole in your chest and sticking something down there close to your heart, with then cords coming out of your shoulder that go to the battery and everything that operates the pacemaker. And as a result of not getting pre-treated with antibiotics and then probably not getting a high enough dose of antibiotics afterwards, he developed septicemia. He went into the hospital Monday during the day. He was delirious. He's still kind of delirious because of it. All because he got a blood infection that was easily preventable via a good antibiotic regimen. If you go to medical school and you get trained, you should be able to look at that person's chart and say, oh, this person's been on Coumadin for the last 5 years. We probably need to take precautions with respect to infection, with respect to blood loss, with respect to all of these other things, because you should be critically thinking well enough that you foresee what the potential problem might be based on the person's history. We had a faculty member who used to work here, and he insisted that medical school students, doctors, are not scientists. And I argued with him quite a lot. Well, doctors are scientists. They're actually getting symptoms from patients. They're creating hypotheses as to what those patients might have in terms of what's wrong with them. And then they do tests to verify or reject those types of hypotheses. And in the same way, doctors are scientists because they should be critical thinkers. And when you don't critically think in the medical field, sometimes things go drastically, terribly wrong. He's probably going to be in the hospital for the next couple of weeks because of all of the complications having to do with what should have been pretty standard procedure. So yes, critical thinking is certainly something that we hope that you're gaining here, but a good place to gain that is actually through the process of doing undergraduate research. And so those are the reasons that we have you do it. All of those reasons that you mentioned, except maybe that it looks good on the transcript. It looks much better to go into an environment and actually be functional and intelligent and brilliant and smart. All right. Today, we are going back to basics. How many of you have had Mendelian sorts of genetics, doing Punnett squares and stuff like this in some other class? High school, college, whatever. Okay, so your hand should be up. We did this in evolution in college.
Oh yeah, we did.
Okay. All right, so everybody's been exposed to this, so we're going to run through this pretty quickly. stop me and slow me down if we're going through it too quickly, because it's still important for you to get, but it should all feel very familiar to you. The first thing I want to do is I want to go back and review the relationship between meiosis and why meiosis is implicated in knowing what to do when you're doing these types of crosses. You should be able to do, by the end of this, be able to do the simplest monohybrid crosses to predict the outcome of inheritance patterns. Recognize the standard outcomes of monohybrid crosses so that in the hopes next week when we come back from spring break, we're going to be doing, we're going to be continuing with this, but we're going to be looking at non-standard outcomes of Mendelian crosses, because that tells us things about whether or not genes are linked on chromosomes with one another. It also tells us about different forms of expression of traits. And so in some cases, a trait is expressed because there's a dosage effect. We've talked about dosage effects in the past. And so having 2 copies of a gene in a homozygote gives you a different phenotype than only having 1 copy of gene if you're a heterozygote, versus having no copies of that gene if you're a homozygous recessive. So recognizing these non-standard outcomes is going to be important. And then using pedigrees, oftentimes students are thrown by how to work through pedigree analyses, and so we're going to teach you that. We're also going to give you some homework. This is going to be the first time that you've gotten homework that you actually have to fill out and do, and the homework that we're going to be giving you is about doing things like figuring out more complicated genetic crosses, doing some pedigree analysis and stuff like that, so that you get some practice doing those kinds of things. So that's what you should be able to do at the end of today and into the week after spring break is over. So remember that genes, we've talked a lot about gene expression, but now we're going to be talking about inheritance. Genes are involved in both of those things. We've already talked about the structure of the genome, and so genes are located at particular positions called loci. These changes in DNA sequences, mutations, result in changes in morphology and phenotype. We're going to talk mostly about diploids because that's what most organisms that you would be normally interacting with are like. We'll note when we get to bacterial genetics and linkage mapping in bacteria, how bacteria differ from diploid organisms in how they do inheritance. It's different enough that they require their own special treatment. So we're going to be mainly talking about diploid organisms, and then we're going to be talking about kind of some conventions for how we label things. And you guys have already seen a variety of different ways of denoting genes. You've seen the way genes were denoted in the Gar et al. Paper. There are simpler and more complicated ways of denoting genes. But back to meiosis. Remember that in meiosis, we start out with diploid parental cell, and we end up with what here at the end of meiosis 1? Are these diploid or are these haploid? It's test 1 material now. They're haploid. So we started off with 6 chromosomes, 3 pairs of homologous chromosomes, and at the end of telophase 1, whoa. Oh, yeah. At the end of telophase 1, we have 2 daughter cells. Each daughter cell has 3 chromosomes. Each of those is one of the 2 halves of the homologs. So we had a blue one, we had a purple one, we have a red one, blue, purple, red. We end up here with blue, purple, red. At that point, we've gone from the diploid state to the haploid state. And then the second division is just like meiosis, but because we've had crossing over back here in prophase 1, what has happened to genes on a particular chromosome? Are those genes all the same now, or have they been altered in some way? Yeah, they've been altered because you have this process of crossing over in prophase 1, such that these genes that are at the tips of the chromosomes will tend to break off and exchange chromosomal segments, such that on a given chromosome, you might have a different combination of alleles of the different genes there than you had before you went through crossing crossing over, so that here at the end, you end up with 4 haploid daughter cells, and the 4 haploid daughter cells have different combinations of alleles than they had when you started this process out. And so this is how you end up getting 4 daughter cells that are all haploid, but they're also all genetically different from one another. They're genetically distinct, rather than being genetically identical. And so this is how we make gametes. Gametes are ultimately what come together to form a zygote. And so gametes are what are carrying the chromosomes that have the genes that determine the phenotype of the offspring. So whenever I go to work out a genetics problem, the first thing that I do if I'm given 2 parents, parent A and parent B, is I ask myself, what kind of gametes can this person make, or this individual make? What type of gametes can this individual make based on what I know of this individual's genotype? What type of gametes can this individual make based on knowing what I know of their genotype? If you can get what their gametes are going to be, doing the cross is relatively easy because then it's just combining all of those possible gamete types. So remember that meiosis results in these gamete types, so if you keep this process in mind, it will help you think through the gamete formation process in doing any simple genetic cross. Questions about that relationship? Okay. You've probably heard about Mendel. Austrian monk, did some experiments back in the 1860s and discovered, voila, the theory of inheritance. He had some advantages over Darwin, who was also trying to crack the inheritance problem at the same time, in that he chose the right kind of organism to use. Darwin was looking at pigeons and talking to pigeon breeders and things like this. But the problem with pigeons is that pigeons don't have a lot of their phenotype determined by these single gene traits. Their phenotype is determined by what we refer to as quantitative traits, traits that are underlain by variation at lots of different loci of different genes. And those become a little more tractable unless you know some statistics, and really the field of statistics hasn't actually been invented at that point yet. And so he chose to work on garden peas. And one of the nice things about garden peas is that they have a number of traits that are governed by single genes, and so they have either or kinds of character distributions. But the other thing that is cool about them is that they have closed flowers. The sexual parts of the flower, so these are all what we refer to as monoecious plants. One house, basically, is literally what it means, a single house. And what that means is that both the male and the female parts are on the same plant. And in this case, they're not only on the same plant, but they're actually in the same flower. And so you have a typical flower structure where you have a pistil. This is where the ova are located. And then you have stamens with anthers on the tip of them. And these anthers produce pollen, which is the male gamete. The ovum here at the base of this pistil is the female gamete. The pollen goes, the pollen has a number of different cells in it. Some of these cells create a tube that grows down through here, and then the one that fertilizes, the cell that fertilizes the ovum follows behind it. And so then you have fertilization. Once the ovum is fertilized, then you're at the zygote stage. And so in some plants, anthers are located on a different plant altogether, and the pistillate flowers are located on a different plant. In some plants like corn, the anthers are at the top and the pistils are at the bottom, about midway down the stalk, so that they're separated spatially even though they're on the same plant. And in some cases, both male and female flower parts are in the same flower. But that's true for a lot of flowers, but only for certain types of flowers do you have them enclosed in the flower in such a way that it's difficult to cross-pollinate from one individual to another individual. And that's the case with pea plants. All of the male and female parts are inside of this internal structure here, and insects and wind and things like that can't get in there to pick up pollen and carry it to another plant, and it's also closed up so that pollen from another plant can't come in and fertilize the ovum. And so in this way, these things tend to self-pollinate. We talked earlier in the semester, and I don't know if you remember this, what happens to a group of organisms if you force them to interbreed generation after generation after generation? What?
Inbreed depression.
Okay, so we can get inbreeding depression, but what's the main consequences of inbreeding?
Spit it out.
Hmm?
Did I say it?
I don't know. Did you say it?
I just said they're all going to eventually look similar. Spit.
They're going to all look similar because when you create what is called a recombinant inbred line, these things become what your book refers to as pure breeding. Why are they pure breeding? They'll look the same. The parents look the same as the offspring. What's the only way in which you have parents looking the same as offspring looking the same as parents?
Keeping the same as their species. Breeding with the same sex species. Same sex species have to breed with each other.
Well, they are the same species. So, I mean, by one definition of species, everybody, only individuals of the same species can breed with one another.
Like they're homozygous dominant? Like
They're not necessarily homozygous dominant, they're just homozygous. Whether it's recessive or dominant doesn't really matter. If you do this over and over and over again, every gene will end up being homozygous. And this is why offspring look the same as the parents, is because the parents don't have any genetic variation. When they go through this process, all of the gametes look all of the same because all of the alleles, there's only one allele type in each of these things. And so all of the gametes end up looking the same, and as a result, all of the offspring end up looking the same. This is what a recombinant inbred line is all about, and this is why you create recombinant inbred lines to use in genetic studies. So I told you about recombinant inbred lines in flies, where they took 1,700 flies and put each female fly in its own vial that produced offspring, and then they kept all those offspring together, and they did that for 25 generations. And at the end of 25 generations, what they found was that for like more than 99% of all loci, they were homozygous at each of those loci. Now each line was differently homozygous from one another, and that gives you a lot of genetic power, because now you know this line is all homozygous, but it's homozygous in this particular way. This other line is all homozygous, but it's all homozygous for all those genes in a different sort of way. And then you can look at what those individual mutations, what those individual allele types are like in terms of expressing phenotypes and expressing responses to stress or whatever it is that you're doing with them. And so essentially, before there was such a term as recombinant inbred lines, Mendel recognized that he had recombinant inbred lines of peas, and they're recombinant inbred lines because they are all just self-fertilizing. There's no outcrossing of one pea plant to another different pea plant. And so this led to what he called true breeding flies, or pure breeding flies, meaning that the offspring look just like the parents because there's no genetic variation at any given locus that you're looking at. So this was good for him because then he could open up the flower as the flower was developing, snip out the anthers so that they wouldn't self-fertilize, and then other plants that had different phenotypes, he could take a paintbrush, brush those anthers, bring that pollen to this flower over here that didn't have any anthers, and force them to cross-pollinate. And so in that way he could do genetic crosses. Once he's done with that, he closes the flower up, and then that prevents any other plant from fertilizing it. And so he could control which plants had sex with which other plants, essentially. And so he could control what he was crossing with what because of the structure of the flower. He also had the advantage in that he had these discrete characters. Flower color, they're either purple or they're white. Flower position, they're either axial or they're terminal. Seed color, they're either yellow or they're green. Seed shape, they're either round or they're wrinkled. Pod shape, it's either inflated or it's constricted. Pod is either green or yellow. The stem is either tall or dwarf. And so we had these discrete categories that were easily assignable to a category in terms of assigning phenotypes to these things. All of the characteristics on the top are the dominant characteristic, and all the ones on the bottom are the recessives. What do we mean when we talk about dominant versus recessive?
In the dominant gene, it's expressed.
Go a little further.
Just kind of seeing what I remember. So like dominant gene, heterozygous, capital A, little a. If it has the dominant allele, it'll be expressed like that.
Okay.
Is that right?
That's actually better, yeah. So a dominant it means that it's expressed if present. Doesn't matter what the other allele is that it's combined with in a diploid organism. If it's present, it's going to be expressed. A recessive allele expressed when?
Well, like another recessive, but it has to be homozygous. Okay.
There's another condition where you get recessive alleles expressed. Yeah, that's already up here, homozygous. I mentioned this in passing some weeks ago, many weeks ago. It's something that happens in sex, it also happens in sex-related traits.
Whenever they're crossed, like whenever one thing's on another thing.
One thing's on another
Like
thing.
it gets switched, like it's not supposed to happen, like in a normal situation.
I'm not sure what you mean by that. In the case of XY chromosomes in humans, what's different about those two?
What do you mean?
No, these have to do with chromosome structures.
Oh, the Y is like small.
The Y is tiny, isn't it? Let's go back and look at something from before.
You know what I'm talking about? I don't know what it's called.
Human karyotype, X chromosome, Y chromosome. Here's where the centromere is, and so in theory they could have drawn this up a little bit so that the centromeres matched. But in this case, this half has a lot of extension beyond that little section on the Y chromosome. This half has a lot of extension beyond this section of the Y chromosome. If you have a recessive allele that's out here at the tip, what's on the other chromosome? Nothing is there because there is no chromosome out there because you got this dinky little Y chromosome here. And so in this case, this is what we would refer to as hemizygous. What does hemi mean? And it's not just a type of engine in Dodges.
Not full.
Not full, or half actually. And so what it basically means is that you have half of it and there's not another half that's there. And so in some organisms, you have sex determination this way because you have an X and a Y, and the Y is actually determining chromosome. It's what we call chromosomal sex determination. In some other cases, it's actually just lacking that chromosome at all makes you male or female, depending on the condition. In which case, whatever is on the chromosome that remains is what gets expressed, whether it's a dominant allele or a recessive allele, simply because there's nothing on the other chromosome to counter it. And so don't forget situations of being hemizygous. This is the easy one. This is the one that you remember from high school. They probably didn't tell you or taught you about hemizygous in high school. Welcome to college. So that's a good way of thinking about these. Functionally, though, at the level of gene expression, what are a lot of recessive alleles then? How do recessive alleles differ from dominant alleles if you were to, for example, look at the sequence of the actual gene? So you have gene structure. Hopefully you all did this correctly on the exam. Structure of a gene for most alleles, this is the top box, the core promoter, exons, introns, blah, 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 basic structure of a gene. When you look at different alleles, it's basically, here's the gene, but there's one form of the gene that is one allele, and there's another form of the gene that is the other allele. Functionally, how do those alleles oftentimes differ when we think about a gene being a dominant expression of a trait or a recessive expression of a trait? They're not, and your book talks about this. Let's go back to the PowerPoint for today. Some of these are simpler to think about than others. What's the difference between a purple flower and a white flower in terms of the phenotype? Okay, but coloration is caused by what?
Light absorbance.
Hmm?
Light absorbance.
Light absorbance, but ultimately what's absorbing the light? So if it's a purple flower, it's absorbing all of the wavelengths except purple, and it's reflecting purple back. So what's doing that absorption and reflectance?
Chemicals that are
What kind of chemicals?
proteins.
Pigments, which are ultimately proteins. So ultimately, to have a purple flower, what do you have? You have the flower producing a purple pigment. That purple pigment happens to be a protein. So how do you get a white flower?
Different protein.
Is it a different protein?
No.
What? You get no protein. So if you have no protein, there's nothing in there absorbing or reflecting light, and so it's just reflecting everything, which is why you get a white flower. So this would represent a gene that is functionally producing a protein, and this is a, your book refers to it as what? What are the kinds of consequences of mutations in, for example, a coding region?
Wait, is it a non-coding gene?
It's just a non-coding gene. So something happens in here. Maybe there's just a wholesale deletion such that the coding sequence is gone in the recessive. Or it could be that there is a deletion or an insertion in one of these exons that leads to a frame shift. So it creates a protein, but the protein ends up not even being a pigment protein. It ends up being some other kind of protein. It doesn't have anything to do with pigmentation. So your book refers to these kinds of alleles as what? Loss of function. Yeah, so these don't have to be, recessive alleles don't have to be loss of function alleles, but oftentimes they are. So the dominant allele, it isn't dominant in the way you normally think of the term dominant. It's just that this produces a functional product. And this represents a loss of function mutation. Now, once again, they're not always going to be like this, but the vast majority of them end up being this in terms of relationship between dominant and recessive. The easiest way to remember that is just think of flower color. Do you produce a pigment or do you not produce a pigment? If you produce a pigment, then the pigment affects the color. And if you don't produce a pigment, then you don't have any pigment molecules in there. It just reflects every wavelength of light there is. Why are plants green? Yeah? Okay. Chlorophyll. What is chlorophyll?
Pigment.
It's a pigment. It's a pigment that absorbs all of these wavelengths of light except green. It reflects green back to the environment. And so that's what pigments do. And the lack of pigment basically means that you get rid of that. If you take a leaf, a green leaf off of a plant, and put it in ethanol for a little while, the pigments in plant leaves are, at least the green pigments, chlorophyll, is soluble in ethanol. The ethanol that it's in will slowly turn green because it's basically leaching all of the chlorophyll out of the plant. And when you pull the leaf out of there, it will be what color? It will be white. Because there is no pigment in there doing this absorption and reflectance. So, true breeding varieties are parental peas producing offspring peas that look exactly like them. Whether that's for seed color or seed shape, it doesn't matter. Round seeds produce round offspring, wrinkled seeds produce wrinkled offspring. But then what Mendel did was he took wrinkled seed plants and crossed them with round seeded plants. And what he found was that in the first generation, strangely enough, and supposedly much to his surprise, all of the offspring were round. What that tells you is that round is the dominant phenotype. The dominant allele codes for round seeds. And the recessive is recessive because in this first generation, the F1 generation, the first filial generation, it recedes to the background. You don't see that trait coming out. But then if you allow those plants to self-fertilize, in the next generation you get round seeded offspring and you get wrinkled seeded offspring. And so you start with round, crossing with wrinkled. In the parental generation, in the first filial generation, everybody is round, but then if you allow them to self-fertilize, in the second filial generation, the F2 generation, you get round offspring again, but the wrinkled suddenly reappear. So he figured out that it wasn't that whatever's causing wrinkled just disappeared. It was always there because it appeared again. So if it was here and here, then it also had to be here, whatever that factor was. And he actually called these things factors. He didn't call them genes. So whatever that factor is that's causing these guys to be wrinkled, it's being hidden somewhere here, and then it reappears here.
Why does it take it next generation?
Well, that was his question. And so he began to think about, well, what are the ways in which this can happen? Now keep in mind, no such thing as chromosomes at this particular time in history. Not much knowledge about cell division and mitosis and meiosis and things like this. But he was a mathematically trained person, and so he thought about this in terms of math. Well, how can you get this happening? Well, you could get this happening if any given trait was governed by two factors. And his thought was that when you are true breeding, you have two factors that are all the same. Oh, let's do this with round. So you have two factors that are the same. And in one case, those same factors give you round. In the other case, the same factors give you wrinkled. If you combine these such that your offspring also have to have two factors, your offspring will have one of each of them. the wrinkled factor is still there, but it's being masked by the other factor, which is the round factor. Now, in this case, I've now interrupted a step here. I always ask myself, what types of gametes can this individual create? This individual can only create one type of gamete. This individual can only create one type of gamete. I always circle my gametes so that I don't confuse them with genotypes, although you can't really do that now because these guys are diploid. When those things come together at fertilization, you have one genotype from every possible offspring will have that one genotype. All of your seeds are round. But if, once again, these factors, these different forms of this factor, are inherited independent of one another, what came to be known as independent assortment, then they basically, this individual can produce one dominant allogamete. It can also produce a recessive allogamete. And we can put these into a Punnett square. And he didn't invent the Punnett square, but basically what you would get out of this is you would get these genotypes. And 3 of these would be the dominant phenotype. And one of these would be the recessive, because this genotype is the same as that genotype up there. And so the only way that he could figure out where you would get this kind of thing, because the other thing that he noticed is that when you allow this thing to self-fertilize, you get some wrinkled seeds, but you get a lot of round ones. Not only that, you get about 3/4 of your seed population shows the round phenotype, and about 1/4 of the population shows the recessive phenotype. And so based on that proportion, he figured it must be governed by 2 factors. These 2 factors get inherited independently of one another. They don't tag along with one another. And as a result, they segregate during gamete formation. And you get this very predictable ratio of 3/4 to 1/4. And we work this out nowadays in a Punnett square. He began with the results of a cross and worked backwards to get to this idea. We now get the idea and we work forwards typically, but he had to start here and ask, how do you get both these phenotypes out of a bunch of round peas that came from round and wrinkled peas, and why on earth are they in these kind of repeatable proportions? And so he studied this with lots of different individual traits, so he did lots of what we call monohybrid crosses dealing with a single trait. And he found this 3-to-1 ratio over and over and over again. And so he had to come up with a way of explaining that 3-to-1 ratio. And this was the concept that he came up with for doing that. So he worked backwards from his results to come up with the underlying mechanism. So, once again, this is the cross that we were doing. Pollen and eggs will all be this. Pollen and eggs will all be this allele. That'll produce the heterozygote, which we call it nowadays. These heterozygotes can produce 2 types of alleles. All possible combinations of those 2 types of alleles are down here. 3 of those will be dominant. 1 of them will be recessive. This is your standard cross. Doesn't matter whether you think of it this way. Doesn't matter whether you think of it this way. It all gives you the same answer. You need to separate genotypic ratios from phenotypic ratios though. The genotypic ratio is that you have 1 homozygous dominant, 1 homozygous recessive, and 2 heterozygotes. These 2 classes here, the homozygous dominant, the heterozygote, will all produce 1 phenotype, the round to wrinkled. And here's the other thing that I want you to get in the habit of doing. In your book, it doesn't really make this distinction very well. But anytime you write a ratio, what should you also be giving me when you write a ratio? Okay, so if this is a phenotypic ratio, what should you be giving me?
Should be going order of dominance recessive, right?
Well, so I would like to know what these are. This is 3 round to 1 wrinkled, because otherwise I might think that this is 3 elves to every 1 dwarf in a battle in Middle Earth. It could be 3-1 anythings to anything. In the same way, if this is the homozygous dominant relative to the heterozygote relative to the homozygous recessive, that's useful to me because guess what? I can write this ratio this way, and it's perfectly correct, as long as doesn't matter. What is this a ratio of? The L4 human component of the Fellowship of the Ring. Is that what I'm looking for when I ask you a question about a genetics problem?
Yes.
No, it's not. Yes, everything's about Lord of the Rings today also. Gandalf is a wizard. It's not really clear that he's human.
Let's say he's not.
I think he is not. I think he would say that he is not. I mean, what other human has the powers of Gandalf? Makes you wonder.
So that would be a 1-to-1 ratio.
So reference your ratio to something. You're making a cake. You have a ratio of flour to sugar. It's important to know which of those 2 numbers in a ratio goes to flour and which one goes to sugar. One of them makes a good cake and the other one makes a hideous abomination. So reference these things when you write them. All right. And who wants a hideous abomination? All right. Now, if you have a plant with a dominant phenotype and you don't know what its genotype is, one of the ways that you can do this is to perform what's called a test cross. If you allow it to self-fertilize, it'll either be true breeding, in which case it will produce what? It'll produce homozygous dominant seeds. They'll all be round, so everything will be round because it can only do this section of the Punnett square because it doesn't have this section of the Punnett square. So if you self-fertilize and it gives you nothing but the adult phenotype, the parental phenotype, you know that it's true breeding, you know that it's homozygous dominant. If it's heterozygous, it will give you 3 round and 1/4 1/4 this, the recessive allele, but the problem with that is detecting that 1/4 is difficult. So wouldn't it be better if you could have a larger proportion of those recessives? Well, the way to do that is to take a true breeding recessive individual that you know is homozygous recessive, back cross it. If this guy is homozygous dominant, what will all of the offspring be? They'll all be round. If, on the other hand, it's a heterozygote, you'll get 2/4 of the population, or 1/2, that will be heterozygous and be round, and you'll get half of the population that will be homozygous recessive. And so you can up your detection probability with a test cross by basically back crossing the recessive phenotype to the individual that was produced up here, and you have a 50-50 chance of seeing wrinkled seeds. And if you see any wrinkled seeds at all, you know that parent had to be a heterozygote rather than a homozygous dominant. And so the reason that this is called a test cross is because it's a way of testing the questions so far? Appropriate rate of speed? Okay, all right, this should all be reviewed to you. You can work with Punnett squares. I actually like Punnett squares. I love Punnett squares in reality. Not everybody relates to Punnett squares, so we can also think about this from the standpoint of what they call a branching diagram, a branching tree. If you want to know what the probability of something happening is, the example that they give you in the book is what's the odds of getting all heads on a flip of a penny, a dime, and a quarter. Well, you have a 50-50 chance of getting a head or a tail on the penny. You have the same chance of getting a head on the dime. You have the same chance of getting a head on the quarter. And so the probability of getting a head on the penny is a half. The probability of getting a head on the dime is a half. The probability of getting a head on the quarter is a half. The product rule basically says the combined probability of independent events is the product of their individual probabilities. So you would have a 1/8 chance of getting 3 heads on 3 flips of different coins. 1/8 is that probability because there's 1, 2, 3, 4, 5, 6, 7, 8 different possible combinations, but we didn't have to work through all of the different possible combinations. All we had to know was what's the probability of head on penny, head on dime, head on nickel, or quarter rather. 1/2, 1/2, 1/2. You can do this with dice as well. It doesn't really matter. If you want to know what's the probability of rolling a 7 on dice, you can get a 7 by getting a roll of 4 on the first die, a roll of 3 on the second die. If we're making things complicated, you could get 3 on the first die and 4 on the second die. You could get 6 and 1, or 1 and 6, or you could get 2 and 5, and 5 and 2. These are all the possibilities. What's the probability of getting a 4 on any given dice? 1/6. What's the probability of getting a 3 on a given dice? 1/6. Probability of getting 2 independent things happening is their product. So there's a 1 in 36 chance of doing this. There's a 1 in 36 chance of doing this, 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 and a 1 in 36 chance of doing that. If you're playing craps and you need a 7 to win, you have 1, 2, 3, 4, 5, 6 different ways of getting that with 2 dice. So what's your chance of winning on any given throw? It's the combination of all of those individual chances. So your chances of winning are 6 in 36, or 1 out of 6 chances. Now, 1 out of 6 chances, I'm not going to be playing craps. Or if I am, I'm going to be playing it for pennies and nickels, not dollars and 20s and hundreds. But if you're into craps, this is your chance of winning on any given throw of the dice. We add these up because we could win by any of these possibilities. And so you add up those different possibilities because it doesn't matter which route you take to getting 7. The important thing is that you got to 7. This is the sum rule. This is the product rule. Now, the cool thing about this is if you do a test cross, if you have a round individual and you don't know what its genotype is, and I'll follow the convention of your book because it's the convention that I've always used. When I don't know what one of the members of a genotype is, I'll just leave a blank there. So this is what I know, this is what I don't know. Some people will write a question mark there. I just leave it blank, your book leaves it blank. If we cross that to wrinkled, our choices for an outcome are basically twofold. One, everybody comes out round, or we get half round and half wrinkled. This only occurs if the genotype of the parent is homozygous dominant. This will occur if the genotype of the parent is heterozygous. So we do the test cross and we get a bunch of offspring. How many offspring do we need to look at before we decide it really is round? Well, we can use these rules of probability to figure this out. If we look at one offspring and it's round, what's the chance of getting one offspring from this cross that's round? Maximum probability. Well, maximum probability is if this guy is homozygous dominant, then the probability is 1, because everybody's going to be round. But if it's heterozygous, what's the probability? It's a half. So at this point, when there's a 50-50 chance that it could be one or the other parental genotype, do we want to make the claim at this point? No. So we look at a second seed, and it is also round. What's the chances of getting 2 round seeds? If this is heterozygous. 1/2 times 1/2, 1/4. If we look at a third one, so now the chances of seeing 2 in a row is considerably smaller than the chances of just seeing 1, right? If the parent is heterozygous. If we find another one that is round, what is the probability of that? Now having 3 round seeds in a row. If we observe a fourth, the probability of that is 1/2 multiplied times the previous, which is 1/16. If we observe a fifth and it's round, that has a probability of 1/2. So the probability of observing 5 round seeds if the parent is heterozygous is 1 out of 32. What's 1 divided by 32? Well, it's 3%. So there's a 3% chance, if this parent is heterozygous, of observing 5 offspring. If you observe 5 offspring and they all happen to be round. If you feel good about that, then you're done. That dude is heterozygous. I'm sorry, that dude is homozygous dominant, because there's a 97% chance that you would have seen 1 wrinkled seed in all of this. So the chances of seeing 5 round seeds, if you have a heterozygous, is 3%, which means that the probability of seeing at least 1 wrinkled out of 5 seeds is 97%. So the probability of doing one thing is equal to 1 minus the probability of the other outcome. And so if you want to put the nail in the coffin, look at 5 more seeds. That's 1/2 raised to the 10th power, which is 0.1 of 1%. The odds of seeing 10 round seeds, if this individual here is really heterozygous, is really, really small. You want to up it, look at 50 of them, and the chances of seeing this are what we would refer to in the literature as vanishingly small. And so you don't have to look at a ton of offspring in a test cross to determine what the genotype of the unknown phenotype is. You only need 5 to 10 offspring to determine that. By the time you get to 10, the odds of not seeing a wrinkled crop up in here if this individual is heterozygous is really, really small. Questions? Have a a good spring break. We'll pick up categories on the Monday after spring break.
No.
That'll be-
No, we don't. We took both of us. We don't have that. I'm pretty sure we don't. I'm not going, so I'm pretty sure we don't.
Luke, did you transfer in? Or did you change?